Hello guys, welcome to Dr. Science. So today we are going to talk about what are apolipoproteins, or we can simply call them as your apoproteins. So this is my second part on the chapter called as your metabolism of lipids or lipid transport and storage, which is taken from your biochemistry textbooks. So please watch my previous video in order to understand this. So I already told you that lipids, in order to get transported into the plasma, they need to contain a special type of protein. Okay, so this is the outer protein, and inside we have lots of lipids. Okay, because lipids are insoluble in water, hence they need a protein covering to get transported. Now this outer protein, there is outer protein, and the inside one is your lipids. Okay, this entire thing is called as your lipoproteins. Now I already told you that lipids are the passengers, whereas the protein shell, the protein covering which is present on the outside, it is your car. There are different cars and different passengers. For each one I am going to discuss about it, don't worry. So this entire complex is called as your lipoprotein. Now this lipoprotein, we have to know the structure of a lipoprotein, right? Now this is a basic structure of a lipoprotein, but in biochemistry textbook it is given, this is the diagram that is given, which is quite complex. So I am going to break it down for you guys. So I already told you that the outer shell is made up of protein and the inside is lipid. Inside the passenger is lipids. So basically lipids are of two types, four major types. Four major types of lipids are present. We will divide them into two subtypes, which are your non-polar lipids and your amphipathic lipids. So what are these amphipathic lipids? What is the meaning of these amphipathic lipids? Amphipathic lipids are nothing but they have a head, okay, and not only that, they are going to have a tail. This head part is called as your polar region or your hydrophilic region. It means it loves being in water, it likes water. Okay, suppose if I place water here, obviously this polar component or the hydrophilic component, it will go and it will be turning that side. Okay, now this tail part, it is your non-polar side. Okay, or your hydrophobic, it means it is water fairy. Now this type of arrangement, where do you see it? This type of arrangement is normally seen in amphibians, right? Amphibians like frogs. So basically frogs, they can live on water, and not only that, they can live on the land, hence they are called as amphibians. Similarly, this lipid is called as amphipathic lipid because it not only loves water, it also hates water, right? Hence this is called as your amphipathic lipids. So what are your two types of amphipathic lipids? The two types of amphipathic lipids are your, so suppose this is a lipid, let me draw this one clearly. So this is a lipid. On this lipid there are phosphates attached on it. So one of the example of our amphipathic lipids are your phospholipids. And not only that, the second type of amphipathic lipids are your cholesterol. So cholesterol and your phospholipids, they are called as your amphipathic lipids. Now what are meant by non-polar? Non-polar means they are hydrophobic, right? They are your hydrophobic lipids. So what are the non-polar lipids? Non-polar lipids are nothing but your cholesterol with esters, or we can simply call them as your cholesterol esters. Now we don't have to forget one of the important one we are going to talk about. Those are your, yes, glycerol. Suppose this is a glycerol component. To this glycerol there are 3 fatty acids attached to it. How many fatty acids? There are 3 fatty acids. This is one fatty acid, this is one fatty acid, and this is one fatty acid. Now this type of arrangement is called as your triacyl glycerols. Okay, glycerol with 3 fatty acids is called as your triacyl glycerols. So the non-polar lipids are your cholesterol esters and your triacyl glycerols. So I already talked that there is in a lipoprotein, the outer covering, let me draw this one clearly. In a lipoprotein, the outer covering is called as your protein shell, whereas the inside there are fatty acids, right? But the fatty acids are not arranged randomly. First there is a monolayer. This monolayer, let me draw this one with a different color. So there is a monolayer of your amphipathic lipids. So what are your amphipathic lipids? It has a hydrophobic and a hydrophilic end. So obviously I'm going to draw a simple diagram here. So here is a head and here is your tail. Similarly, like this, there is a monolayer of your amphipathic lipids. This is your monolayer of amphipathic lipids. So what are your amphipathic lipids again? The amphipathic lipids for your, yes, phospholipids and cholesterol. And after that innermost layer, now you may ask that why there is a monolayer of amphipathic lipids present outside. Because suppose if there is water here, imagine there is water here, so this head is the hydrophilic end, hence the layer is arranged in such a manner the water is exposed to the hydrophilic end, right? And the non-polar lipids, they are hydrophobic lipids, hence they are present innermost. They are your hydrophobic lipids. There is a layer of hydrophobic lipids innermost, or the core. Inside the core, these are your non-polar lipids. So what are the examples of non-polar lipids? The answer is simple. Those were your triacyl glycerols, or we can simply call them as TAGs, and cholesterol with esters, cholesterol esters. Okay, so once again, so lipoprotein, it is consisting of a protein shell, which is the outermost component, and it is consisting of a lipid layer. The first lipid layer is your amphipathic lipid layer. So there is a monolayer of amphipathic lipid layer. There is a monolayer, means single layer of amphipathic lipids. And after that, the innermost core is consisting of non-polar lipids such as your TAGs and cholesterol esters. Okay. So I will just show you this diagram. So as you can see here, this image is taken from your biochemistry textbook. So I already told you there is a core of mainly non-polar lipids. So these are your non-polar lipids. All these randomly arranged things are your non-polar lipids. Non-polar lipids include your cholesterol esters and triacyl glycerols. Okay. Now there is an amphipathic layer. As you can see here, there is nicely arranged head, and this is the tail. There is head, this is the tail. There is head, this is the tail. This type of arrangement are called your amphipathic lipids, right? And what are the two types of amphipathic lipids? Those are your cholesterol, which is here, and not only that, your phospholipids. Okay. Now the outer this outer layer, thick layer, this is the protein shell. Now you may be wondering, sir, what is this apoprotein? What is this integral apoprotein? What are this apo-B, apo-C, apo-A? Okay, now I will dig deeper. So I already told you there is a protein shell, and inside there is a lipid which needs to be transported, and because of lipids are insoluble in water. So this protein is having certain types of moieties. Okay, so there are the outer protein shell, it is containing a protein moiety. Okay, this protein moiety of a lipoprotein, the entire complex is called as lipoprotein, right? And the protein moiety of this lipoprotein is called as your apolipoprotein, or we can simply call them as apoproteins. Okay, they are your apolipoproteins or your apoproteins. Now you may be wondering, sir, what is apo-B, apo-C, apo-E? So I'll be okay, my pad is a little stuck. So I already told you the protein moiety of a lipoprotein is called as your apoprotein, and the apoproteins are of different types. There are different types of apoproteins, and each apoprotein, the distribution, this statement is taken from your biochemistry textbook, so I want everyone to pay attention. The distribution of apolipoproteins characterizes the lipoprotein. So what is the meaning of this? The distribution of apolipoproteins. Apolipoproteins are nothing but the protein moieties, right? Characterizes the entire lipoprotein. Lipoprotein is the entire complex, right? So suppose we are taking liver here, example. Okay. So this is the liver, and this liver is producing a substance called as your very low density lipoprotein. Very low density lipoprotein is a type of lipoprotein. Okay, each and every lipoprotein, there are a lot of lipoproteins, like lot of cars and passengers. I already told you what are different types of lipoproteins in my previous video, so please watch that one in order to understand this one. So liver is producing a substance called as your very low density lipoprotein. Suppose this is the protein moiety of the lipoprotein. Suppose this is a protein moiety, and inside the very low density lipoproteins, okay, carry mainly lipids. The major lipid here is your triacyl glycerols. Okay, very low density lipoproteins and chylomicrons, they mainly transport lipid called as triacyl glycerol, and this very low density lipoprotein is consisting of a protein moiety. Suppose this is the protein moiety, and this protein moiety is called as your ApoB100. Okay, so what can we say that when you see this type of picture? So I will give a simple example. So for India, the phone number will start with +91, and for Philippines it is +63. For UK, the phone number will start with a universal code called as your +44, and for USA it stands, I think it's +11. So basically by seeing this we can say that it is coming from India. The phone number is from India, the phone number is from Philippines, the phone number is from UK and USA similar. So if a lipoprotein is having ApoB100, we can say that it can be a very low density lipoprotein, or very low density lipoproteins can break down into a complex called as intermediate density lipoproteins, and eventually they can be converted into low density lipoproteins. So all these three will be having ApoB100. It means they are having a stamp, okay, they are having a stamp on them called as ApoB100. And when we see this stamp on them, when we see this ApoB100 protein present on them, we can confidently say that these proteins are nothing but your VLDL, IDL or your LDL. Because, or I can give you a similar example. Basically the Indians which are present in the southern part, they are brown to black in color, right? And the English for us also is kind of weird compared to the American people. Whenever you see an Indian, you can easily find out that this guy is coming from India. Similarly, these proteins are like this. If there is this marker present on them, we can immediately say that these proteins are either VLDL, IDL and or LDL. So that is the meaning of this one, the statement which is given in your biochemistry textbook, which is the distribution. Okay, my iPad is kind of stuck. Okay, so this is the statement that is given: the distribution of apolipoproteins characterizes the lipoprotein. It means the apolipoproteins are your markers, and if these markers are specific for each and everyone, and this characterizes the lipoprotein, it can say what ethnicity you are. Okay. So I would like to give a few examples of such lipoproteins. So basically I already told you that liver produces a substance called as your, so suppose this is the liver, and liver produces a substance called as your VLDL, and VLDL, suppose this is a protein part, this protein part is going to have ApoB100. Now ApoB100 is not present on the outside, okay, it is present on the inside. You will be hearing apoprotein A or apoprotein C or apoprotein E. They are present on the external surface, whereas ApoB is always an integral protein. Okay, ApoB is an integral protein, hence it cannot be removed so easily. So if VLDL is okay, so if this lipoprotein is having ApoB100 present on it, we can confidently say either it is a VLDL, and VLDL can eventually get broken down into intermediate density lipoproteins, and intermediate density lipoproteins can break down into low density lipoproteins. All these on their surface will be having apoprotein or ApoB100. Okay, this is very important because it is given in your biochemistry textbook. So next we will talk about HDL. HDL is a good cholesterol, right? HDL, it carries your good cholesterol. Now what is the lipoprotein that is carried by your high density lipoprotein? So high density lipoprotein, this is the protein moiety, and this protein moiety is going to have ApoA. Whenever you are having ApoA A protein, apoprotein A, we can 100% say that it is a high density lipoprotein. Okay, so very low density and intermediate density and your low density lipoproteins, all they are going to have is B100 present on them, whereas in the case of HDL, which is carrying your phospholipids inside HDL, it is carrying mainly phospholipids and cholesterol. So high density lipoproteins, they will have ApoA. So whenever you see a protein with ApoA, you can confidently say that it is a high density lipoproteins. Obviously chylomicrons are also having ApoA, but HDL is far greater than your chylomicrons. Now what about chylomicrons? Chylomicrons are the transporters or lipoproteins which are chylomicrons or is a lipoprotein which is coming from your small intestine or your GIT, right? Lipoprotein from GIT. So what is the apoprotein present on chylomicron? The apoprotein which is present on the chylomicrons are nothing but your, this is the integral protein again, is your ApoB48. Now sir, what is the difference between ApoB48 and ApoB100? Actually the apoprotein B, normally suppose this is one amino acid, this is second amino acid, third amino acid, and eventually all the amino acids will fuse and make a long chain called as your protein. Okay, so this apoprotein is having nearly, apoprotein B100 is having nearly 4536 amino acids, or we can say it is having 100% amino acids, but the ApoB48 is only having 48% of amino acids present on it, hence the name it was given as ApoB48. Okay, so I am going to take a small test on you guys. Try to answer this one. So I am going to draw an a lipoprotein. So this lipoprotein is going to carry ApoB100. Can you name what is this lipoprotein called? Yes, those are your very low density lipoprotein, or your intermediate density lipoproteins, or your low density lipoproteins. So I am going to give another one. So here is the lipoprotein, and this lipoprotein is going to have a stamp, and this stamp name is ApoB48. Can you name the lipoprotein? Yes, the lipoproteins are your chylomicrons. Okay, and finally the last one. So this is a lipoprotein. On this lipoprotein there is an external protein, and this protein is called as your ApoA protein. Can you name the lipoprotein? Yes, it is your high density lipoprotein. So that was about your lipoproteins. Thank you guys.